Hello friends, welcome back, and now we are going to start the topic of enzymes. We are moving on with the topic called enzymes. Okay, let's start with enzymes. What do you mean by enzymes? So what are enzymes? What are enzymes? Okay, so let's see what do you mean by enzymes. Enzymes are proteins. Mostly they are proteins with exception of ribozyme. We'll see later. The ribozymes are also enzymes, but they are not proteins. Ribozymes are RNA. Ribozymes are RNA with catalytic activity. We'll keep mentioning the RNA which are ribozymes when we encounter them. So basically they are RNA with catalytic activity. All right. So this is the chemical structure of most of the enzymes. For this discussion today, when we are talking about enzymes, we will assume that enzymes are proteins. We will not mention them as ribozymes. We will assume that all the enzymes that we're talking about are proteins. So what is the action of enzymes? They act as the biocatalyst. They act as a biocatalyst. They decrease the energy of activation. They decrease the energy of activation of a reaction. At this point, what you have to remember, uh, the decreasing of energy of activation is the process. The result, rate of reaction increases. In other words, endpoint is achieved faster. So you should also know what do you mean by endpoint. What is the endpoint? I'm saying the endpoint is achieved faster, but what is the endpoint? The endpoint is equilibrium. Equilibrium is achieved faster. But at the same time, a very important point to keep in mind is although the rate of reaction increases, although the equilibrium is achieved faster, the concentration at equilibrium is same as for an uncatalyzed reaction. So whether you use the catalyst means the enzyme or not, the reactions of substrate and product at the equilibrium will be the same. Whether we have the catalyzed reaction or we have the uncatalyzed reaction, the concentration of the substrate and product at equilibrium is going to be the same. Okay, I can't see anyone in there. Can you just send me your thumbs up so that I can see how many people are currently present with us? Just give me a hi or a thumbs up so that I get an idea how many people are currently logged in with us. Anyone? Do we have anyone online? Let's see. Okay. So now that you know a little bit about the chemical structure, let's talk about the classification of the enzymes. Classification of enzymes is a broad concept which is very frequently tested. Details, very much details are not required, but you should have a broad idea of the classification. Now here I'll mention the classification. The current classification has been given by, very good, International Union of Biochemists. Great. International Union of Biochemists Sometimes to this we also add the term MB, that stands for molecular biology. So International Union of Biochemists, or you can also add the term MB, IUBMB, IUB or IUBMB. The classification given by the IUBMB has four parts. We focus only on the first part. The other parts are beyond the scope of the MBBS syllabus, so we don't go into the other parts. We only mention the first part of the classification, which is the class. Otherwise we have the class, subclass, subclass, and the function. So we don't go into the other details. We focus on the first part, which describes the class of the classification. So this classification will have two things. One is the name. The second will be the number. Each class also has a number. So oxidoreductase is a class 1. It is involved in oxidation and reduction. So either there is a gain or loss. Either there is gain or loss. Gain or loss of electron, proton, or oxygen. Gain or loss of electron, proton, oxygen. This will be the oxidoreductase. This will be the oxidoreductase. Some subclasses that you should know about, these include the oxidase, the oxidase, oxygenase, oxidase, oxygenase, dehydrogenase, the peroxidase. These are some names which you should know about. The oxidase, the oxygenase, the dehydrogenase, and the peroxidase. Another very important name would be the hydroxylase. Hydroxylases are also oxidoreductase. Hydroxylases are also oxidoreductase. So if you see any of these terms, you should know that you are talking about the class 1, that is the oxidoreductase. Then we have the class 2. Class 2 are the transferases. They'll mostly end up with the transferase term only. So transferases, they will transfer functional group from one place to another. The class 3, the class 3, the class 3 are the hydrolase. Hydrolase catalyze hydrolysis through water. In other words, in very simple words, they will break bonds. They will break bonds using water. They will break bonds using water. And the good example is the digestive enzymes. Most of the digestive enzymes, for example, amylase, trypsin, chymotrypsin, peptidase, endonuclease, exonuclease, all of these will come under the category of hydrolases. Hydrolases catalyze the hydrolysis through water. They will break the bonds using water. Class 4 are the lyses. They act on double bonds. They break bond without water. They break bond without water. Now, in the new classification, large number of synthases large number of syntheses belong to the class lyase. Large number of syntheses belong to the class lyase. All right. So lysases will act on double bond. They break the bond without using the water. Then we have class 5, isomerase. Some other terms that we've already seen in addition to isomerase are mutase, the epimerase. These are also isomerases. Either it ends with isomerase. For example, we had seen the phosphohexose isomerase, triose phosphate isomerase. We had also seen the mutase and epimerase we saw in the galactose metabolism. In galactose metabolism, we had seen the epimerase. So all of these are isomerases. Then we have class 6. Class 6 is ligase. Ligase will join two molecules. So important subgroups are synthetases. All synthetases require ATP. All synthetases require ATP and they are ligases. Similarly, carboxylases. Very good. One of our friends has given the name racemase. This will also come to isomerase. So carboxylase is also a ligase. Now, I'm sorry. Oops. Okay, so as of 2018, we have a new classification published in August September by the International Union of Biochemists, and that is translocase. Previously, translocases were considered the transport proteins. They were considered as a transport proteins, and they were not classified as enzymes. They were not classified as enzymes. But as per the latest nomenclature, the translocases will also be listed as enzymes. For example, we had seen the ATP ADP translocase. So they will shift molecules from one compartment to another. This is the latest addition to the classification. So we have the term a translocase. Translocase is class 7. It will shift the molecules from the ATP ADP translocase shifts the ATP from mitochondria to the cytoplasm. The ADP is shifted from cytoplasm to the mitochondria. So translocases will shift molecule from one compartment to another. This is the new addition and very likely to land up in your exam. Translocase is a very new addition and it is likely to land up in your exam. Please make note of the translocases and you should be clear about the translocases. So now that we have done the classification, let's look at the constituents of the enzymes. We broadly classify the active enzyme that is the holoenzyme into two parts. The protein part, which is the apoenzyme. The protein part, which is the apoenzyme, it is the active part, the place where the catalysis occurs. So it is also known as the catalytic unit. So apoenzyme is the protein part. And then we have the non-protein part, which are subdivided based on the type of attachment, whether the attachment is covalent or the attachment is non-covalent, depending on the type of attachment, whether it is covalent or it is non-covalent. So if it is attached non-covalently, depending on a type of molecule, whether it is organic molecule or inorganic molecule, we have the terms coenzyme and cofactor. Coenzymes are mostly shuttles. Mostly shuttles. They'll bring in the co-substrate for the pathway. They generally bring in the co-substrate for the pathway, whereas the cofactors are mostly activators. For example, metals, the magnesium, the calcium, the iron, and the manganese, they come in to activate the enzyme. So we call them the cofactor. If the group is attached covalently, it doesn't matter whether it is the organic, inorganic, we call all of them as the prosthetic group. All of them are called as prosthetic group. They may be organic, they may be inorganic, it does not matter. Okay, so this is about the constituents or the subgroups of the holoenzyme. The final functional enzyme is known as the holoenzyme. Now, depending on the attachment of metals, we can call the enzymes differently. If the metal is present as the cofactor, we call it the metal activated enzyme. And if it is present as prosthetic group, we call it the metalloenzyme. We call it the metalloenzyme. So, metal activated enzyme and the metalloenzyme. So first we talked about the general the nature of the enzyme. Then we talked about the classification. Then we talked about the subgroups or the constituents of the enzyme. Let us talk about the properties of the enzyme. These properties will help us to describe the enzyme, and we are able to fully understand the function of an enzyme. The first part of this is the active site. The first part of this is the active site. When I say active site, immediately we get to know three things. What do you get to know? The substrate which is attached, because the substrate will attach at the active site, so we can see the substrate which is attaching. The catalysis will occur at the active site, and the product will also be formed at the active site. So active site can immediately tell us three things: the substrate, the catalysis that is occurring, and the product which is going to be formed. We use a combination of either substrate and catalysis or the product and catalysis to give the name of the enzyme, because just by looking at the name we'll be able to describe the function, the primary function of the enzyme. So we use this combination to describe the name of the enzyme. For example, if I say DNA polymerase, so here the catalysis is polymerization and the product is DNA. Similarly, if I change the name and I give you the name RNA polymerase, so immediately you know that polymerization is happening and the product is going to be the RNA. So just by looking at the name, we get to know few details about the enzyme, which particularly describes the active site. The active site has the property of attaching the substrate. This is where the product will be formed via the catalysis. Then we talk about the catalytic efficiency, which is described by the two parameters, the Vmax and the Km. The catalytic efficiency is described by the two parameters, the Vmax and Km. I'll go to a different page to show you the Vmax and Km. When I say a Vmax, what does it mean? When I say Vmax, what exactly does it mean? So please note, Vmax is initial velocity under ideal conditions. So the second question becomes, what do you mean by ideal conditions? The VI under ideal conditions, VI stands for initial. So we always talk about initial velocity only. We always talk about the initial velocity only. So VI under ideal conditions. What are the ideal conditions? The ideal conditions are optimum pH, the optimum temperature, but last, the most important, infinite substrate concentration. The infinite substrate concentration is a very important feature of the ideal condition. So the Vmax is described as the Vi under ideal conditions, which includes optimum pH, the optimum temperature, and the infinite substrate concentration. Please note, that you cannot have infinite substrate. You can have an approximately infinite substrate, means the substrate is not limiting, but you cannot have infinite substrate. So what this means, we can calculate the Vmax, but you cannot measure the Vmax. You can calculate the Vmax, but you cannot measure the Vmax. So how do we identify or look at the catalytic efficiency in the lab? So what we do, we look at the graph which will give us a clear picture. We look at the graph which will give us the clear picture. Okay, on the x-axis we have the substrate concentration, on y-axis we have the initial velocity. So we have the value called the Vmax. We have the value called the Vmax. Okay, we have the value called the Vmax. We cannot measure the Vmax. Why? Because if you look at the graph, the plateau phase will meet the Vmax when the substrate concentration becomes infinite. So we cannot actually measure the Vmax, we can calculate the Vmax. So what we do, we calculate the Vmax and then we calculate something else. We calculate another parameter. This parameter is Vmax by 2. So first we calculate Vmax and then we calculate the Vmax by 2, and then we drop the perpendicular from here on the graph, and then we drop the second particular on the x-axis. Wherever this perpendicular touches the x-axis, we label this as Km. So if you look at the description of the Km is substrate concentration at Vmax by 2. Km is the substrate concentration at Vmax by 2. So we can measure the Km and it will tell us, it will give us an idea of the catalytic efficiency. But what exactly does Km tell us? I have told you this previously. The Km tells us about affinity. It tells us about affinity, and affinity is inversely related to Km. Means if we have more affinity, we will have less Km. If we have less affinity, we will have more Km. See, if the affinity is high, the initial rate of reaction will be more. Let's say the final Vmax is the same. If the affinity is less, the initial rate of reaction will be less. Let's say the final Vmax is the same. So look at the Km in these conditions. When high affinity is there, the Km is less. When low affinity is there, the Km is more. So Km is inversely related to affinity. The Km is inversely related to affinity, which you have to keep in mind. This is a very, very important point, and we'll come across this point again and again in our discussion. Don't forget this. Affinity and Km are inversely related. Affinity and Km are inversely related. So we use the parameters, the Vmax and Km, to describe the catalytic efficiency. We use them to describe the catalytic efficiency. The third part of the property that you have to describe is specificity. All enzymes do not act on random substrate, they will act on a very specific substrate. This is the specificity. So here what you have to know, there are two models which describe the specificity as shown by the enzymes. They are lock and key model. The lock and key model tells us the enzyme catalytic site and the substrate are exactly complementary. They are exactly complementary, there is no deviation. And the second one is the induced fit model. The induced fit model says the catalytic site where the substrate will come and attach is slightly flexible. Once the substrate comes, this site can modify itself, it can modulate to properly attach to the substrate. So, the lock and key model and induced fit model. If you have to choose one, go with induced fit model, but please remember both are partly correct. If you have to choose one, in that case go with induced fit model, but remember the lock and key model is also not incorrect, however it is not able to describe some of the relationships. Okay. Okay, so the three very important properties of the enzymes, the active site, the catalytic efficiency, and the specificity, they give us a very good idea of what type of enzyme are we dealing with. We get to know the reaction, we get to know the Vmax, we get to know the Km, we get to know how strongly or weakly it is attaching to the substrates, and what type of relationship is it there. Is it going to act on only one substrate, or it is going to act on multiple substrates? Okay, in addition to this, there are some more points which may or may not be seen. Means we call them additional properties. They are not essential. They may be there, they may not be there. The first is allosteric site. Allosteric simply means allo means other, and steric means site. The term itself means site. So it is a site other than the active site. The allosteric site means a site other than active site, meaning molecules can come and attach here. Molecules can come and attach here. If the enzyme has the allosteric site, we call it the allosteric enzyme. If the enzyme doesn't have the allosteric site, it is a normal enzyme. If it has the allosteric site, we call it the allosteric enzyme. So the molecules will come and attach. Once they attach, what they do? They will modify the catalytic efficiency. This means they can be activators also and they can be inhibitors also. They can be activators as well as inhibitors. If they are activators, they will improve the Vmax, they will decrease the Km. If they are inhibitors, they will decrease the Vmax, they will increase the Km. So depending on the type of molecule, whether it is the activator or it is the inhibitor, the changes in Vmax and Km will occur. So we use the term modify. We don't say increase or decrease, we say the catalytic efficiency is modified. Then we have covalent modification. Here, certain molecules or groups attach covalently to the enzyme. They attach covalently to the enzyme. In allosteric site, the molecules generally attach non-covalently, whereas in the covalent modification, the molecules will generally attach. Generally, what they by definition they will attach covalently. The net result is the same, meaning they will modify the catalytic efficiency. So when the molecule is attached to the allosteric site or there is a covalent modification, both of them will result in change in catalytic efficiency, that is the change in the Vmax and the Km. Lastly, some enzymes can require the cofactors or coenzymes for the optimum activity. They will come and help the enzyme to carry out the catalysis better. So these are some of the very critical properties of the enzymes which you should know. They generally come as statements where you have to identify whether the given information is right or wrong. So these are some conceptual points which you should know about the enzymes. Once you know the properties, we will now talk about the enzyme kinetics. Now enzyme kinetics is a very fundamental question, very frequently asked in your exam. Let's look at enzyme kinetics. The enzyme kinetics, just like for any catalyst, is described as zero order, the first order, the second order. zero order, first order, or second order. Please note, in this case we are mostly focusing on the first order kinetics. We are mostly focusing on the first order kinetics, and the equation which very well describes the first order kinetics is the Michaelis-Menten equation. Because it is one of the best equations which describes enzyme kinetics, you should be able to identify the correct Michaelis-Menten equation. You should be able to identify the correct Michaelis-Menten equation, which says the initial velocity. You see, again I'll highlight, it talks about Vi, the initial velocity, is equal to Vmax into substrate concentration divided by Km plus substrate concentration. So initial velocity is equal to Vmax into substrate concentration upon Km plus substrate concentration. We have already seen what this graph looks like. On x-axis we have substrate, on y-axis we have velocity, initial velocity, and this is a hyperbolic curve. We've already seen hyperbolic curve. Okay, we already know what is the Km. Km is the substrate concentration at which the velocity of reaction will be half of the maximum velocity, at which the velocity of the reaction will be the half of maximum velocity. Now, the Michaelis-Menten equation is a very good equation if we want to study a single reaction, but it becomes problematic when you are trying to compare different reactions. When you're trying to compare different reactions, in this case, the Michaelis-Menten equation is not very good. Why? Every time you want to compare at a given point, let's say we want to compare at this point, you will notice that each of the graph has a curve at that point. So for comparing the change in velocity, what you need a tangent here, you need a tangent here, and you need a tangent here also. So first you need to draw the tangent before you can do the comparison, which is a very cumbersome process. So we don't want to do this. So what we do, what we end up doing, whatever is on the left side we take it to the right, and whatever is on the right we take it to the left, right? In other words, it becomes 1 upon Vi is equal to Km plus substrate concentration upon Vmax into substrate concentration. When you simplify this changed values, when you simplify this changed values, what you get is known as the Lineweaver-Burk plot or the double reciprocal plot. So remember, the Lineweaver-Burk plot itself is derived from the Michaelis-Menten equation. We have not changed any parameters in the Lineweaver-Burk plot. They are exactly the same as for the Michaelis-Menten equation. Only thing we have done is we have interchanged the left and the right side. But now it becomes very simple. Why? Because we can compare this to a very well-known equation, the equation for a straight line graph. This means when we plot the Lineweaver-Burk plot or when we draw the double reciprocal plot, every time we are going to get a straight line graph. Every time we are going to get the straight line graph. Look at this. Every time we get the straight line graph. Why do we call it the double reciprocal plot? Because on y-axis we have 1 upon velocity, the reciprocal of velocity. On the x-axis we have 1 upon substrate, the reciprocal of substrate, and that is why we use the term the double reciprocal plot. We use the term double reciprocal plot described by the Lineweaver-Burk and therefore known as the Lineweaver-Burk plot. So what should you know in the Lineweaver-Burk plot? Number one, you should know what is the slope of the graph, that is Km upon Vmax. Where the graph cuts the y-axis, where the graph cuts the y-axis, we call it the y-intercept. Where the graph cuts the x-axis, we call it the x-intercept. So these are the three terms which you have to know: the slope, the y-intercept, and the x-intercept. Okay, so in the line of Burke plot, the slope, the y-intercept, and the x-intercept. X-intercept is -1 upon km, y-intercept is 1 upon Vmax, and the slope is km by Vmax. Okay, look at the graph. The graph is very good. The graph is very good uh, for comparison of the different reactions. The comparison of the different reactions can be done very well with the help of graph. But very rarely, even this graph will fail. See, we have a line going like this. We have another line going like this. And we have another line going like this. So what is happening? Multiple lines are crossing in the same area. Multiple lines are crossing in the same area. This is known as clustering. So when there is clustering, at that time the Lineweaver-Burk plot will fail. We cannot use it for comparison if there is clustering on the Lineweaver-Burk plot. Otherwise, it is a very good tool for comparison of different reactions, except when there is clustering. So what do you do when there is clustering? When there is clustering, we go to a second set of reactions known as the single reciprocal plots. They can be used to minimize clustering. They can be used to minimize clustering. Please note down the names and the parameters on the x and y axis for the two plots: the Eddy-Hofstee plot and the Hannes-Wolff plot. So these are the two plots which we can use when there is clustering. Otherwise, we prefer to use the double reciprocal plot for comparison of equations or reaction, and we want to study a single reaction. In that case, we will use the Michaelis-Menten equation. Now that you know the double reciprocal plot, we can start talking about inhibitors. But one last point which you need to know, that is the factors affecting a reaction velocity. Once you know this, then we can quickly go and start talking about the enzyme inhibition. So what are the factors which affect the reaction velocity? First, substrate concentration. We already seen it gives us a hyperbolic curve. The second would be the pH. See, we said for enzyme activity there is an optimum pH at which you find the Vmax. At optimum pH you find the Vmax. But if you move away from this pH either to the left or to the right, there is a sharp drop in the performance of the enzyme. There is a sharp drop in the performance of the enzyme. We call it the bell-shaped curve. We call it the bell-shaped curve, right? The same is true for temperature also. For temperature also, you'll get the bell-shaped curve only. Okay, so for pH and temperature, we get the bell-shaped curve. And the last factor in the list, and that is enzyme concentration. What happens to the velocity if you change the concentration of the enzyme? And the answer is perfectly linear relationship. The relationship is perfectly linear for enzyme. You increase the enzyme, the velocity will increase. You decrease the enzyme, the velocity will decrease. So these are the effects of the different factors which affect the reaction velocity of an enzyme. So we'll list them together very quickly. We'll list them together. Okay, let's list them very quickly. Temperature will give us the bell-shaped curve. The pH will also give us the bell-shaped curve, and the enzyme concentration will give us the linear curve. The enzyme concentration will give us the linear curve. So these are the factors affecting reaction velocity. Now we'll move on to the enzyme inhibition. These are the different graphs that we have drawn. Now we're moving on to the enzyme inhibition. Now when you talk about inhibitors of enzymes, we can have two class of inhibitors or two class of inhibition. It can be reversible or it can be irreversible. These are the two primary classes, reversible and irreversible. The two primary classes, reversible and irreversible enzyme inhibition. All right, they are further subdivided into three subclasses. Here you only need the names and examples. The reversal one are subdivided into further three classes, but here you need names, example, the change in Vmax, the change in Km, the point of attachment, the point of attachment, and the change in the Lineweaver-Burk plot. Why? Because the management of the reversible inhibitors will be different, will depend on the subtype. Whereas the management for all the irreversible inhibition will be same. That is a symptomatic management. So we'll take a look at them in the same detail. For the reversible inhibition, we'll talk about all these parameters. But for the irreversible inhibition, will only give the examples and will mention the subtypes. The details of the irreversible inhibition will not be required at this stage, so will not go into them. So first we are talking about the reversible. We are talking about the reversible inhibition. Three subtypes: the competitive, the non-competitive, and the uncompetitive. The example of competitive is malonate versus succinate. Malonate versus succinate acting on the enzyme succinate dehydrogenase. We had seen this when we were doing the TCA cycle. I had mentioned the malonate as acting on the succinate dehydrogenase. It is a competitive inhibition. The non-competitive inhibition, lead, acting on ferrochelatase, aldehyde, both of them are enzymes of heme synthesis. Both of them are inhibited by lead. The uncompetitive, the example is alkaline phosphatase inhibited by the phenylalanine. The alkaline phosphatase inhibited by the phenylalanine. Okay, so what I'll do, what I'll do here, I'll very quickly. Okay, let me complete this inhibition, then I'll come back to this question. I have a question from Akshay, I can see. I'll take that question. Let me complete the inhibition and then just remind me to take up that question. Okay, I'll take up that question. Don't worry. Okay, so the three subtypes of inhibition, we have the competitive inhibition, the non-competitive inhibition, and the uncompetitive inhibition. Right. So here, first I told you you have to tell us about the change in the Km, the change in Vmax, the point of attachment, the point of attachment. These are three main points that you must be able to provide. So in competitive inhibition, the Km is increased, meaning the apparent affinity decreases, but the Vmax will remain normal. The Vmax will remain normal. In non-competitive inhibition, the Km is normal, but Vmax decreases. In uncompetitive inhibition, both decrease. The Km also decreases, the Vmax also decreases. Please note this is paradoxical, means the apparent affinity increases. This is paradoxical, but you have to remember that in uncompetitive inhibition, the Km decreases. Km decreases. Let's look at the point of attachment. In competitive inhibition, the point of attachment is the catalytic site. The point of attachment is the catalytic site. In non-competitive inhibition, the point of attachment is the allosteric site. There is no competition for attachment at the active site. Rather, the molecule is attaching at the allosteric site and it will modify the Vmax and the Km. In uncompetitive inhibition, it isn't attached to the allosteric site. It isn't attached to the catalytic site. Where does it attach? It attaches to the enzyme substrate complex. So it waits for the enzyme substrate complex to form. Once the enzyme substrate complex is formed, only then the uncompetitive inhibition is going to act. The uncompetitive inhibition will act only when the enzyme substrate complex is formed. Now, the question very commonly will be image based in these type of situations. It will not be a tabular question or a straight line question. Rather, it is likely to be the image based question. You will be given the different types of Lineweaver-Burk plot, where you will be asked to identify what type of inhibition are you looking at, or maybe there can be a second step question: this type of inhibition is seen with which type of drug. So it can be a two-part question. We can mix the pharma with the biochemistry there, so it can come like that also. So the challenge will be to identify the Lineweaver-Burk plot and what type of inhibition is being shown in the graph. To do that, you have to remember a very simple thing. And what is that? You have to look. You have to look at what is normal. Vmax is normal in which condition? in competitive inhibition. If Vmax is normal, then we are looking at competitive inhibition. If Km is normal, then we are looking at non-competitive inhibition. And if nothing is normal, in that case we are looking at the uncompetitive inhibition. So this is how you can quickly identify the graphs. If the Vmax is normal, competitive inhibition. If the Km is normal, the non-competitive inhibition. If both are modified, in that case we are looking at the uncompetitive inhibition. I'll show you in the graph so that it becomes more clear. This is the normal graph. You can see this is the normal uninhibited graph. We have on the 1 upon Vmax and minus 1 upon Km. These points are there. I'll write them for reference. This point is minus 1 upon Km, and this point is 1 upon Vmax. Okay, so let's look at the three panels and then the picture will become clear. Look at these two graphs. This is the normal and this is the inhibited graph. They are overlapping at this point. This point denotes the 1 upon Vmax. If both of them have same 1 upon Vmax, this means they are going to have the same. They are going to have the same Vmax. And what is the condition when you have the same Vmax? The answer is competitive inhibition. The same Vmax is seen in a competitive inhibition. Okay, look here. In second graph, they have the same minus 1 upon km. If they have the same minus 1 upon km, they are going to have the same km. So in this case, the km is unchanged. If km is unchanged, then obviously we are looking at the non-competitive inhibition. We had mentioned this previously. If the km is normal, we are looking at the non-competitive inhibition. In the last equation, the Vmax for both is different. The km for both is different. That is why it is definitely the uncompetitive inhibition. It is definitely the uncompetitive inhibition. If you know and understand this, how to use the normal value to identify the different type of Lineweaver-Burk plot, then your work is done. It is very very simple. You can quickly correctly identify the different types of graphs. I hope the graph is clear to everyone. Everyone is able to understand the graphs. I hope everyone is able to understand the graphs. If there are any doubts or confusion in the graph, let me know. I'll draw the graphs independently and I'll show you. If there are any doubt or confusion in the graphs, please let me know, because they are likely to land up in your exam as image based questions. So you should be very very clear about the graphs that we have just discussed. Okay. There are no doubts about the graphs, I hope, so that I can move on to the next topic. I can move on to the next part, not the next topic, next part, and that is the irreversible inhibitors. The irreversible inhibitors. I told you here you only need to know the names and examples. The details of the irreversible inhibitors are not required. So please note them. I'll talk a little bit about the last one, that is suicide inhibitor. It commonly lands up in the exam, particularly for examples. So first you note this down and then I'll quickly talk about the suicide inhibitors. So, like I said, the irreversible inhibitors are also of the three subtypes. The irreversible inhibitors are also of three subtypes. The first subtype, the group specific reagent. The group specific reagent, example molecule DIPF, it acts on a chymotrypsin and the acetylcholine esterase. Target enzymes chymotrypsin and acetylcholine esterase. Second name, the reactive substrate analogs, also known as the affinity labels, also known as affinity label. All right. So the 3-bromoacetol phosphate acting on the triose phosphate isomerase, the TPCK acting on the chymotrypsin. These are some examples. The last example, which is very important, is that of suicide inhibitors, also known as the mechanism based inhibitors. Okay, I'll talk a little bit more about these suicide inhibitors because they land up in your exam. So, what do you mean by suicide inhibitors? The first point is the inhibitor is consumed, in other words, destroyed during inhibition. Normally, in the process of inhibition, the inhibitor is not consumed. Normally, the inhibitor is not consumed, but here, what we are seeing, the inhibitor is consumed or destroyed during inhibition. A metabolite binds irreversibly to active site. A metabolite binds irreversibly to active site. And remember, this is a type of irreversible inhibition. The enzyme cannot be reactivated. I'll repeat, the enzyme cannot be reactivated. So, how will the enzyme activity reappear? The enzyme activity reappears only after new enzyme synthesis. This is true for each of the three irreversible inhibitions. This is true for each of the three irreversible inhibitions. The enzyme activity will reappear only after a new enzyme is synthesized. So for suicide inhibitors, remember, the inhibitor is consumed or destroyed during inhibition, meaning it must attach at the active site. Metabolite binds irreversibly to the active site. This also implies that inhibitor is similar to substrate. Inhibitor is similar to substrate. When you talk about suicide inhibitors, the inhibitor is similar to substrate. Now, in the question, you'll find in the slide, you'll find certain examples are already written. I hope you have noted down these examples. The DMPA acting on monoamine oxidase, the penicillin acting on a glycopeptide transpeptidase. I'll give you some more examples. The DMPA acting on monoamine oxidase, penicillin acting on the glycopeptide transpeptidase. Some more examples I'll give. DMPA acting on monoamine oxidase, penicillin acting on glycopeptide transpeptidase, then aspirin acting on cyclooxygenase, commonly known as the COX, commonly known as the COX. The aspirin acting on cyclooxygenase, commonly known as the COX. Allopurinol acting on the xanthine oxidase. Allopurinol acting on xanthine oxidase. The 5-fluorouracil, commonly written as 5-FU, acting on thymidylate synthase. In addition, last but not the least, allopurinol, it is a secondary target, orotidylate decarboxylase. Orotidylate decarboxylase. So these are examples of suicide inhibitors, where the mechanism that we just discussed at the top, the inhibitor is getting consumed or destroyed during inhibition, and therefore it must attach to the active site. One of the metabolites binds irreversibly to the active site, and the enzyme activity will reappear only when a new enzyme is synthesized. Because this is an irreversible inhibition, the enzyme cannot be reactivated. Now, one follow-up question which is asked regarding allopurinol, and which you must know, the metabolite which attaches to xanthine oxidase. What is the name of the metabolite which attaches to the xanthine oxidase? This you must know. This is asked, so you should be able to give this answer. The name is oxypurinol, also known as alloxanthine. The oxypurinol or alloxanthine. Any questions so far? Any questions so far? Anything you want to ask? Any questions so far? I'll take Akshay's question. If there are no other questions, I'll take Akshay's question. Okay. So Akshay asked, sir, enzyme has to attain Vmax, so enzyme concentration should also give hyperbolic curve. So please understand, when we talk about the factors affecting reaction velocity, when you talk about factors affecting reaction velocity, it is assumed all other factors are optimum. It is assumed that all other factors are optimum. All right. So this means at any given ऊपर से बंद. At any given concentration, at any given concentration of enzyme. At any given concentration of enzyme. For that concentration of enzyme, we are already at Vmax. For that concentration of enzyme, we are already at Vmax because the substrate concentration, the temperature, and pH would already be optimum. They would already be optimum. At any given concentration, the other factors will already be optimum. So if I draw the graph for Vi like this, and we see the Vi at this point, if you see the Vi at this point, it is the Vmax for that enzyme concentration. It is the Vmax for that enzyme concentration. So I hope that solves your problem, Akshay. That enzyme has to attain Vmax, so enzyme concentration should also give hyperbolic curve. No, we are talking about the relationship of velocity with enzyme concentration. Enzyme is the worker. If you increase the number of workers, the amount of work done will keep increasing, provided the work is there, and the work will be there because the all other factors are optimum. The substrate is infinite. When we are trying to measure the factors affecting reaction velocity, the other factors have to be optimum, so the substrate will be infinite. You keep increasing the enzyme, more and more substrate can get converted to product, so the velocity, the initial velocity, will keep increasing as you keep increasing the enzyme concentration. So this is the point to keep in mind. This doesn't hold true for others because they are one of the three factors to describe the Vmax. Okay, this is applicable only for the enzyme concentration because it is not one of the three factors used to describe the Vmax. When we describe Vmax, we talk about a fixed enzyme concentration. Akshay, is your question clear? Can you just give me a reply whether you have understood or not? Yeah, very good, very good. Done. Done. Okay, so now that point is clear, some extra points that I want you to know. And there is something called ping pong reaction. Ping pong reaction. This means one or more product is formed before all the substrates will enter the reaction, and classically it is described for the process of transamination. It is classically described for the process of transamination. So I'll show you how it is happening.